Hello there, how are you? Welcome to another biology class. Today in part 3 we will talk about the genetic blueprint of organisms called DNA. Before we start, make sure you have subscribed to this channel and please be kind to like and share this video. Alright then, let us begin.
Sense and antisense. A DNA sequence is called a sense sequence if it is the same as that of a messenger RNA copy that is translated into protein. The sequence on the opposite strand is called the antisense sequence. Both sense and antisense sequences can exist on different parts of the same strand of DNA, that is, both strands can contain both sense and antisense sequences. In both prokaryotes and eukaryotes, antisense RNA sequences are produced, but the functions of these RNAs are not entirely clear. One proposal is that antisense RNAs are involved in regulating gene expression through RNA-RNA base pairing. A few DNA sequences in prokaryotes and eukaryotes, and more in plasmids and viruses, blur the distinction between sense and antisense strands by having overlapping genes. In these cases, some DNA sequences do double duty, encoding one protein when read along one strand, and a second protein when read in the opposite direction along the other strand. In bacteria, this overlap may be involved in the regulation of gene transcription, while in viruses, overlapping genes increase the amount of information that can be encoded within the small viral genome.
Supercoiling. DNA can be twisted like a rope in a process called DNA supercoiling. With DNA in its relaxed state, a strand usually circles the axis of the double helix once every 10.4 base pairs, but if the DNA is twisted, the strands become more tightly or more loosely wound. If the DNA is twisted in the direction of the helix, this is positive supercoiling, and the bases are held more tightly together. If they are twisted in the opposite direction, this is negative supercoiling, and the bases come apart more easily. In nature, most DNA has slight negative supercoiling that is introduced by enzymes called topoisomerases. These enzymes are also needed to relieve the twisting stresses introduced into DNA strands during processes such as transcription and DNA replication.
Alternative DNA structures. DNA exists in many possible conformations that include a DNA, bDNA, and zDNA forms, although only bDNA and zDNA have been directly observed in functional organisms. The conformation that DNA adopts depends on the hydration level, DNA sequence, the amount and direction of supercoiling, chemical modifications of the bases, the type and concentration of metal ions, and the presence of polyamines in solution. The first published reports of DNA X-ray diffraction patterns and also bDNA used analyses based on Patterson functions that provided only a limited amount of structural information for oriented fibers of DNA. An alternative analysis was proposed by Wilkins et al. In 1953 for the in vivo bDNA X-ray diffraction scattering patterns of highly hydrated DNA fibers in terms of squares of Bessel functions. In the same journal, James Watson and Francis Crick presented their molecular modeling analysis of the DNA X-ray diffraction patterns to suggest that the structure was a double helix. Although the bDNA form is most common under the conditions found in cells, it is not a well-defined conformation but a family of related DNA conformations that occur at the high hydration levels present in cells. Their corresponding X-ray diffraction and scattering patterns are characteristic of molecular paracrystals with a significant degree of disorder.
Compared to bDNA, the DNA form is a wider right-handed spiral, with a shallow, wide minor groove and a narrower, deeper major groove. The form occurs under non-physiological conditions in partly dehydrated samples of DNA, while in the cell it may be produced in hybrid pairings of DNA and RNA strands, and in enzyme DNA complexes. Segments of DNA where the bases have been chemically modified by methylation may undergo a larger change in conformation and adopt the Z form. Here, the strands turn about the helical axis in a left-handed spiral, the opposite of the more common B form. These unusual structures can be recognized by specific zDNA binding proteins and may be involved in the regulation of transcription.
Alternative DNA chemistry. For many years, exobiologists have proposed the existence of a shadow biosphere, a postulated microbial biosphere of Earth that uses radically different biochemical and molecular processes than currently known life. One of the proposals was the existence of life forms that use arsenic instead of phosphorus in DNA. A report in 2010 of the possibility in the bacterium Phi1 was announced, though the research was disputed, and evidence suggests the bacterium actively prevents the incorporation of arsenic into the DNA backbone and other biomolecules.
Quadruplex structures. At the ends of the linear chromosomes are specialized regions of DNA called telomeres. The main function of these regions is to allow the cell to replicate chromosome ends using the enzyme telomerase, as the enzymes that normally replicate DNA cannot copy the extreme 3' ends of chromosomes. These specialized chromosome caps also help protect the DNA ends, and stop the DNA repair systems in the cell from treating them as damaged to be corrected. In human cells, telomeres are usually lengths of single-stranded DNA containing several thousand repeats of a simple tag sequence. These guanine-rich sequences may stabilize chromosome ends by forming structures of stacked sets of 4 base units, rather than the usual base pairs found in other DNA molecules. Here, 4 guanine bases, known as a guanine tetrade, form a flat plate. These flat 4 base units then stack on top of each other to form a stable G-quadruplex structure. These structures are stabilized by hydrogen bonding between the edges of the bases and chelation of a metal ion in the center of each 4 base unit. Other structures can also be formed, with a central set of 4 bases coming from either a single strand folded around the bases, or several different parallel strands, each contributing 1 base to the central structure.
In addition to these stacked structures, telomeres also form large loop structures called telomere loops, or T-loops. Here, the single-stranded DNA curls around in a long circle stabilized by telomere-binding proteins. At the very end of the T-loop, the single-stranded telomere DNA is held onto a region of double-stranded DNA by the telomere strand disrupting the double helical DNA and base pairing to one of the 2 strands. This triple-stranded structure is called a displacement loop or D-loop.
Branched DNA. In DNA, fraying occurs when non-complementary regions exist at the end of an otherwise complementary double strand of DNA. However, branched DNA can occur if a third strand of DNA is introduced and contains adjoining regions able to hybridize with the frayed regions of the pre-existing double strand. Although the simplest example of branched DNA involves only 3 strands of DNA, complexes involving additional strands and multiple branches are also possible. Branched DNA can be used in nanotechnology to construct geometric shapes.
Artificial bases. Several artificial nucleobases have been synthesized and successfully incorporated in the 8-base DNA analog named Hajimoji DNA. Dubbed S, B, P, and Z, these artificial bases are capable of bonding with each other in a predictable way. S, B, and PZ maintain the double helix structure of DNA and be transcribed to RNA. Their existence could be seen as an indication that there is nothing special about the 4 natural nucleobases that evolved on Earth. On the other hand, DNA is tightly related to RNA, which does not only act as a transcript of DNA but also performs as molecular machines many tasks in cells. For this purpose it is to fold into a structure. It has been shown that to allow to create all possible structures at least 4 bases are required for the corresponding RNA, while a higher number is also possible, but this would be against the natural principle of least effort.
Acidity. The phosphate groups of DNA give it similar acidic properties to phosphoric acid, and it can be considered as a strong acid. It will be fully ionized at a normal cellular pH, releasing protons which leave behind negative charges on the phosphate groups. These negative charges protect DNA from breakdown by hydrolysis by repelling nucleophiles which could hydrolyze it.
That is all for today, folks. One last thing. Before you go, make sure that you have subscribed to our YouTube channel and be kind to like and share this video. This video is edited by Brooke Solomon, MSc in Genetics. We will see you in the next video. Goodbye.